Hello, welcome to the DNA workshop. Today you're going to play detectives and you're going to learn how applications of DNA technology are used to solve crimes. So let's start in a very cheerful, friendly sort of way. Look at these biscuits. Are they the same?
No.
All right. They're different?
Yes.
A little bit different. Now let's say you wanted to make these biscuits, but you had absolutely no idea how to begin. Do you know how to make biscuits? Not really. Okay, so what would you need? I'm not talking about ingredients. What else would you need if you want to make some biscuits?
Recipe.
Yeah, a recipe. So here's a recipe book for making biscuits, cookies, and so on. And if I look inside, there's maybe 300 different recipes in here to tell us how to make different types of biscuits. And these recipes are written in English, and there's 26 letters and numbers as well, to give us about 300 different ways to make biscuits. Now let's look around at the class here. Let's see, who've we got? Is he different from this young lady here?
Yep.
Bit like the biscuits, eh? So do you think there's a recipe inside them to make them the way you are, and all the rest of you as well? There's a recipe inside you. Yeah, but it's not a recipe in a cookbook, is it? It's a recipe that's inside your cells. And it's not written in this same language as the English language, it's written in the language of DNA. And DNA only uses 4 letters, or the 4 letters we use as abbreviations for chemicals. And over there on the board, you'll see I've got the letters up representing the chemicals of DNA, and you'll see that T is always opposite A, G opposite C. That's a whole nother story about DNA structure. We won't go into that now. You don't even have to know what they're called, but it's adenine and cytosine and guanine and thymine. That's what makes up DNA. I sometimes say to people, do you know what DNA stands for?
I know.
Yes, you know.
Deoxyribonucleic acid.
That's good. If you didn't know, I'd say DNA stands for do not ask. And so we don't have to be threatened by what we're going to do here with DNA today. So, okay, the recipes inside the cells in your body. I like to use a box like this to represent a cell. And there's 100 million cells making you up. And I wonder who's going to be brave enough to lend me one of their 100 million cells this morning. You are, right, good. That's nice. Now, DNA is a long, thin strand, actually, and if I was to come and break open one of the cells on your thumb, so lend me your thumb, hold on to this, and hold on to that. Now I'm going to pretend I'm pulling the DNA strand out of just one cell in your body, and this is how much DNA comes out of just one cell in your body. 2 meters of DNA. Now, if you would be kind enough to lend me another cell, obviously, let's take the wire over there now, pass it along to your friend over there. Now I've taken out another 2 meters, so hold on to that. Maybe pass it over there, stretch it out a bit, take it back down over there. And so you see that's just out of 2 cells in your body. If I was to break, you were very generous, but you probably don't want me to break you down to nothing. But if I did, I'd have enough DNA to wrap around the world from you 600,000 times. That's how much DNA is in your body. And that's the stuff that we're going to look at today. Okay, you can let that go. Okay, so if we could stretch this DNA from here to the sun, it would weigh as much as a paperclip. It's incredibly fine, thin stuff. And in all of us, we have the same DNA in every single cell, but your DNA is slightly different from your DNA, slightly different from your DNA. And it's those differences that are used in forensics, which is using information in legal cases, in court cases, to identify people. And that's what we're going to look at today. For example, any living material after death will still have DNA inside there. And good sources of DNA in crime investigations are blood, for example. Here's some blood. And I want you to listen to this, because later I'm going to ask you to act out a crime scene. So there we go. blood at a crime scene, blood on a weapon. Sometimes, tragically, the only thing remaining, skeletal remains, and so skeletons sometimes are used to extract DNA for forensic investigations. And it's the little differences in the G, A, C, and T that you see up on that board between one person and another that is the basis for making a DNA profile or a DNA fingerprint, which you probably see on programs like CSI. So I'm going to show you today how it all works. A really good source of DNA, got one here, is teeth. Yes, because they haven't been destroyed by breakdown by bacteria in the soil so much. So there's a good source of DNA. Teeth. And those are real human teeth, by the way. And one would grind them up and get DNA out of it for experiments or for working in the laboratory. Now, the DNA that we've got here, as I've discussed, is kept safe inside cells, inside what we call the nucleus. So let's pretend this is a cell. We're going to break it open and get the nucleus out of it. So this represents the nucleus, and the DNA is inside there. I'll put this out of the way till later. And I always like to say how important the nucleus is in the DNA. We're going to have to get DNA out of cells just now and break the nuclei open. And when this nucleus is very nice, this flashes to tell us how important the DNA is. So that's what we're going to be doing just now. It's not going to be me talking, it's going to be you working. Now, I could get DNA by pulling some hairs off your head. Oh, look, he winced. And then we'd put them in a little tube. Here's a little tube. And then we could start getting DNA out. Or I could go over to you. I haven't picked on you yet, have I? Give me your hand. I could take your hand, I could roll up your sleeve, and I could take some blood out of you. But you might not like that at all. I might scratch you and get some of your cells to work on. But let's take DNA out of a plant. Plants don't complain. At worst, green stuff oozes out of them. And the type of plant we're going to use is a wheat plant. And a very good source of DNA I can get from ground up wheat. So that's what we're going to use to extract DNA. And you've got the things to do it down here, and I'm going to go back behind my kitchen counter to show you how to go about it, and then I want you to extract DNA. So as I said, we're going to take DNA out of wheat. So I've got here wheat seeds. As I mentioned before, if we were extracting, say, from bone, we'd first of all have to grind up the bone. We should be grinding up the wheat grains in a mortar and pestle, for example, like this. But that's rather hard work, so I made it a little bit easier for us. I've got ready ground wheat. It's actually called wheat germ, but this germ means the same as germinate. It means the center of the wheat seed. It's a wonderful resource for us because inside the packet is just what we need. Lots and lots of broken up cells with DNA and nuclei. And the starch has been used to make flour, and the bran's been used in all-bran flakes, for example. So you've got a little jar on your tables. Yes, there's the wheat, the ground up wheat. And you've got, here's mine, you've got a teaspoon, and take 1 teaspoon of ground wheat and put it in the jar on your table. And the next thing we need to do is add 3 tablespoons of water. That will be 45 mils. That's about how much we need. So 3 tablespoons of water into your jar. And next you need to stir it. Use the kebab stick, use the flat end of the kebab stick and start stirring. I'm going to ask you, why are you doing this? What did I hear? Yes, because I told you to. But I've told you to for a good reason. Can you see what's happening? Is it dissolving? No, not like sugar in tea. It's not dissolving. We're making something called a suspension. What we want to do is get all the nuclei spinning round and round in the water in this jar, so that when we add the next ingredient, it's going to work on all the nuclei. Now the next ingredient I want you to add is dishwashing liquid. And you can add half a teaspoon of dishwashing liquid. You've got it in these little containers. So put it all in. You can rinse it off if you want to. Put the spoon aside. Now you can start stirring again. I want you to stir gently. Why do you think you'd need to stir gently? What happens if you wash up dishes roughly? Yes, they break. Yes, and you get a lot of bubbles as well. We don't want a lot of bubbles, and we certainly don't want to break up our DNA, which is a very fragile little molecule. Okay, what differences can you see in the liquid in your jars? Yes, yeah, I heard you say that's right. Gloopy, thick, slimy. Yes, slimy. In fact, if it looks like snot, that's great. But scientifically, we might say it's viscous. Yes, it's highly viscous now. I wonder why that is. What do you use dishwashing liquid for? Wow, I'm surprised. Even the boys know. Yes. You use it for washing dishes. And what sort of dirt does it really get off those dishes?
Grease.
Grease, yeah, grease, fat. Now, I want you to throw me that nucleus, but I probably won't be able to catch it. Throw it gently then. Throw me the nucleus. Oh, right. So here's the nucleus. And the nucleus, the DNA, those long threads are inside it, and it's surrounded by something we call a membrane, like a fatty sac is around it. Now, as we said, dishwashing liquid will get fat off things. So if we add dishwashing liquid to nuclei, and now I'm going to have a big magnified nucleus here. So here's my nucleus now, so imagine what I'm going to do. Oh, oh, right. I add dishwashing liquid to it, and it's as if I just popped it. And what has come out of it? What is this representing? Yeah, that's the DNA. So that's the thick, gloopy stuff that you've got in there is DNA. So even though it's so long and thin, you can see it now because there's thousands and thousands of nuclei in that teaspoon of wheat germ that you added. But it's still in solution. We need to get it out of solution. Now, DNA is insoluble in alcohol. Well, like I say, sometimes DNA maybe prefers alcohol to water, like some people do. So we're going to add some alcohol to it, and the DNA will precipitate in the alcohol at the interface where the two meet. We don't want to mix the two. The type of alcohol we'll use is methyl alcohol, methanol, methylated spirits. In the laboratory, we'd use ethyl alcohol or ethanol. But that's more expensive. It works just as well with methanol. But remember, meth is poisonous. Please don't drink it. We need to add an equal amount, an equal volume. So in this tube I've got here, I'll be adding about that much of methanol. And I'll tip it on the side when I do it and run it down the side. I'm just going to give it a good stir before I do that to make sure that all the DNA is there. At the interface, because the DNA will start precipitating from the interface here between the methanol and the water layer. So here we go. Let's see what sort of DNA preparation I've got out of this. Here we are. Oh, beautiful. So you see those white fluffy strings there that are getting pulled up by the bubble? Those are strings of DNA being precipitated into the methanol solution. Now, if you were working in a laboratory, you would be doing basically the same sort of thing. So you might have small blood sample, like I showed you down there just now, or you might have hairs from someone's head pulled out at the crime scene, cells that have fallen off, and then we work in tiny little tubes like this that we call Eppendorf tubes. So we're not working in big jars like you're doing. We use a purified form of dishwashing liquid, but it does the same thing. It emulsifies fat, and we use ethanol, ethyl alcohol. And once we've reached this stage and got the DNA at this stage, we take it and we clean it up a bit more. And after that, it's ready for analysis in the forensic laboratories, just like you see on CSI. So what you can do actually at the end of a workshop like this is to use your kebab stick, fish out some of that DNA and put it in the little vials that you've got there, top up with methylated spirits, and then it will keep forever. Or else you could take a piece and just rub it on the black card that you've got there and dry it down. You see you've got some black paper as well, so you can also see it that way. So you can do that now. You've extracted DNA. In a forensic lab, that would now be analysed using pretty state-of-the-art apparatus. What we're going to do now is learn how it would then be interpreted. But before you can learn how to interpret the DNA profiles, you've got to have a lesson in genetics. So I'm coming around here to do a quick lesson in genetics, at least the part of genetics that you need to know. The 2 meters of DNA that's in every cell is divided up into 46 little pieces called chromosomes. And the chromosomes exist as 23 pairs, chromosome 1 to 22, and the X and Y chromosome, which is the 23rd pair. And I'm going to show you how, when sperm or egg are formed, there's a reduction in the number of chromosomes so that they can repair afterwards. And that is the explanation for why I am wearing one red shoe and one blue shoe. Now, you might think I'm a mad professor, which I probably am, or that I got up in the dark this morning and got dressed, which also happened. But what I want you to think about my shoes as is chromosome pair 6. Because that's the size of my feet. And you can think of the other 23 chromosomes, or other 22 chromosomes ranged around here. So we'll have 1, 2, 3, 4, 5 there. There's 6, 7, 8, 9, 10, all the way out to chromosome pair 23. Now, in a woman, in the ovaries, when the eggs are made, this is meiosis, which you might have learned about in biology. When the egg is made, the first thing that happens is that in the cell that's going to become an egg, they make copies. So now we've got 2 pairs of red and 2 pairs of blue. But after that, only one of each of these chromosomes will go into the eggs. So from now, we would finish up with 4 eggs in the ovaries. And at some stage in development, let's say, one of these chromosomes finishes up in an egg. And let's say that it's this version of my chromosome 6. I should probably tell you that these colours of my shoes are actually representing small changes in the sequence of DNA, those letters that you see there. There's actually 3 billion of those letters amongst these 23 chromosomes, but that's a story I'm going to tell later in bioinformatics. But you just need to know that there are slight differences in sequence, one from the other here, and you inherit those differences from your parents. So this red shoe that I have here is actually representing a copy of a chromosome that I inherited from my mother, and the blue shoe is representing a copy of this chromosome that I inherited from my father, and all the way back through the generations. And I'm showing you what's happening only with chromosome pair 6, but the same thing's happening with the other 23 chromosome pairs. So let us say that this version of chromosome 6, the one that I got from my mom, is the one that finishes up in an egg. Now, if we're going to have a child created from this egg, we need a partner. And I don't have another pair of coloured shoes, but fortunately socks work really well too. So I want this to represent chromosome pair 6 in my partner. And this is 2 versions, one that he inherited from his dad and one that he inherited from his mom. And they're slightly different from each other. So when the sperm is being formed, again, these divide and make replicates of themselves. So we now have this choice in the father. And they go into 4 different sperm cells. And let's say that this is the lucky sperm that swims along and fertilizes this egg, boop, and now we've got an embryo there that's going to develop in the child of this couple. Now, scientists don't talk about colors when they talk about these differences. They give them numbers. The numbers are called alleles. And it's like a genetic identifier. It's my own personal genetic ID. So we can call the red shoe, version of chromosome 6, 1, version 1, version 2, version 3, version 4. And now, look, the children have always got to have half from their mom and half from their dad. And each time it's a 50-50 combination of which they will get, and each of the other chromosomes are behaving independently of each other as they also go into this egg or into this sperm. That's Mendel's laws, but we're not going to go there today either. So if this is a 1-2 and this is a 3-4, what is the genetic identification number of this child? This child's a?
1, 3.
Yes, a 1-3, right, thanks. And now, maybe they have another child, and it will work out totally independently, like a flip of a coin each time. But for the learning purposes, let's say that the version of chromosome 6 that goes into the egg that makes the next child is that one, and the version of chromosome 6 from dad is that one. So what will be the numbering then of that child?
2, 4.
Yes. So you see, each member of the family has their own genetic identification number. And people always say to me, yes, but what if? But they might have got the same. But I'll stress again, all the other chromosomes are also doing their own thing, going into the egg and the sperm. So this is the way we identify people, and this is the basis of DNA profiling, which is one of the applications of DNA knowledge in biotechnology. I'm going to go over to the board there and set up a family, so you will see how this works. So here we have a mom and a dad, and I'm going to give them their genetic identification numbers, their genetic profile. I'll use the same ones as I used just now. Mom will be 1, 2, so we're talking about just one of her chromosomes. Pretend it's chromosome 6, and dad will be 3, 4. And now they have their first child. What would you like me